Hi guys, welcome to lesson 9. So about last lesson, I kind of went way too fast to the point that I may have thrown at you guys 4 hours worth of information in the span of 90 minutes. And I put too much information in that one lesson that we might actually end this course early. So, we'll see. Anyway, welcome to lesson 9. Firstly, we're going to talk about some stuff from the previous lesson that I pushed or rushed over and may have not clarified very well. Firstly, point out the organelles that were taught in the previous lesson. Right, all right. So here you can see, here's a plant cell again, and then here's the animal cell. If you guys recall, to make you remember, a plant cell is more boxy. Do you remember why? What organelle is in a plant cell that makes it more boxy than an animal cell? I'll give you guys like 30 seconds and then I'll answer it myself because nobody's here to actually answer it live. Okay, in the meantime, I'll keep talking and then I'll say what the answer is in later. All right, so first of all, we're gonna review the organelles and what they do because it's pretty important. You'll be tested on them a lot in Science 10. First of all, here we see the purple thingy. That is the nucleus. And then inside that purple thing is the nucleolus where ribosomes are made. And ribosomes are like tiny things. Right? They're tiny organelles that make proteins, which is very important for your cells. The nucleus as a whole, inside it contains DNA, which is like information about your cell. It is the brain of the cell. Around the nucleus, we have the endoplasmic reticulum, two kinds. We have the RER, rough one, and then the smooth one, SER. The rough one, I'm pretty sure it acts as a highway for proteins where ribosomes made in the nucleolus and protein stuff just travels through the endoplasmic reticulum to places. Smooth endoplasmic reticulum, it helps detoxify and sometimes helps break down some stuff, I think lipids, and does some storage. Farther outside of that, we have the Golgi apparatus. Think of it again as the post office where it packages and ships out stuff to around the cell. Then we have the lysosome, tiny dot things. Think of Lysol, they clean up after the cell, they digest your waste. Lysol, they clean up. Ribosomes, oh, I talked about that already. Vacuoles. All right, if we remember, in plant cells, there's only one giant vacuole or few giant vacuoles, but in animal cells, many tiny ones. The role of vacuoles is to store water and food. And then we have the mitochondria. These things. They are the powerhouse of the cell. They make energy. Remember the phrase, powerhouse of the cell. Next we have chromatin. That's not important. Nucleolus, I talked about it. Let's see, nuclear envelope. It's just the skin of the nucleus. Nuclear pore, they're just pores of the nucleus. Oh, wow. Centrioles, they are only in animals and they are in charge of how your animal cell reproduces, how they split themselves in half. Let's see, cytoplasm. I didn't talk about that very much. So cytoplasm is just all the liquid, like a gel-like substance within your cell in general. The role of a cytoplasm is to kind of hold these organelles in place and they give the organelle its shape. They just fill in all empty space in the organelle and it's mostly made of water. And then of course, the thing I mentioned about what makes a plant have a boxy shape, the cell wall, which gives a plant its structure. Yeah, since plants don't have bones, they need extra structure with their cells. And then something that both have is the cell membrane, which is just the skin of the cell. What else do I need to talk about? Ask them for differences. Well, none of you are here, so whatever. Okay, comparing plant, animal, and bacteria cells. This is also something you'll get tested on, probably. Firstly, what we know about plant cells is that you need to remember that they are the biggest cells. Out of all of these, they're the biggest cells. Again, they have a boxy shape. They have chloroplasts, which means they can perform photosynthesis. Unfortunately, we don't have chloroplasts, so we can't photosynthesize. We really should have evolved to have chloroplasts. It's really not fair. Anyway, large central vacuole, whereas animal cells have many small vacuoles, and then plants have a cell wall. And we don't because we have bones, which is not as cool as cell walls, because then if we get stepped on, we die, but plants don't die when they get stepped on, so we should get cell walls. But then we have bacteria, which is special. They are the tiniest cells. They are unicellular, typically, which means they are made of only one cell, whereas animal and plant cells we are a combination of multiple cells. Bacteria also has a cell wall, which is most of them do. They are typically oval shape. I should bring out a picture of a bacteria. Here, let's grab a bacteria cell. Bacteria cell. Here, yeah, look at that. It's an oval shape with a tail. They don't always have tails. A lot of them do. A lot of bacteria have cilia, which means they're hairy, essentially, and then flagella, which is the tail. It helps them move around. This is their legs, essentially. Oh, wrong one. They have a nucleoid region instead of a nucleus, where instead of having all the DNA in a circle, their DNA is kind of just scattered in their cell. And they have few membrane-bound organelles. So you can see here, all functions are within the cell are very clearly defined by different organelles. Chloroplast does this, the mitochondria does this, there's an organelle here, an organelle there. Whereas in a bacteria cell, their organelles, they're kind of just spread. They don't have specific organelles. Everything in them kind of does everything. That's the best way I can explain it. All right, moving on. Cell transport. The cell membrane, we talked about this before. So the cell membrane, we refer to it as a phospholipid bilayer. In the cell membrane, I need to stop doing that. In the cell membrane, they have a phosphate head, which is these circle thingies. And the phosphate heads, which is the circle thingies again, on the cell membrane, are polar, which just means they have a slight charge. They might be slightly positive or negative charge. I forget. And it's attracted to water. So that means the phosphate head is hydrophilic, which just means attracted to water. Here, let me redefine this. Phosphate heads are polar. And polar means the object has a slight charge. Polar objects are hydrophilic, which means polar objects are attracted to water. Next, lipid tails. There are two lipid tails attached to each phosphate head in the cell membrane. They are nonpolar, meaning they have no charge, and nonpolar objects are hydrophobic, which means they are afraid of water. So if you look here in the cell membrane, the sides that face out are the phosphate heads, and the sides that face inside, kind of in the cell, is the lipid tails. Why? Well, your phosphate heads are attracted to water or not afraid of water. And if you remember, there's cytoplasm, and actually your body, a lot of the environment's pretty watery. Your body is pretty watery, there we go. Like all the cells inside your body will be in a pretty watery environment. However, because the lipid tails are like, no, ew, I hate water, disgusting, they face inside so that they don't touch the water that's on the outside. Now, and when there's one phosphate head and there's two lipid tails, one of these thingies that are called a phospholipid. Whoa. As you can see in the cell membrane, there are two layers of phospholipids. So the cell membrane is referred to as a phospholipid bilayer. In the cell membrane, there's also a protein channel, which is these tubes. The protein channel is where diffusion occurs. So stuff that needs to go in and out of the cell goes through these protein channels through passive transport. There are also proteins that are on the membrane or embedded into the membrane. So like these things. I think I explain what they do later. Pretty sure I do. Oh, now we talk about diffusion. So if you guys remember what passive diffusion is when a solute follows a concentration gradient going from high, so a solute is a solid in a substance. I hope you guys remember what a solute is. Anyway, a solute is a solid. Wow. You know what, I give up on trying to define it. Basically, passive diffusion, when objects are moving through maybe a membrane, there we go, some kind of barrier, they don't need any help, they can just move through. But then there's facilitated diffusion. This is a type of diffusion or transport that doesn't need any energy, but it does have some help in the diffusion. So it's like regular diffusion, but uses a carrier protein. So if you see here, this is a carrier protein, that is a protein channel. Carrier proteins allow a solute to go into it, it closes up, and it opens on the other side, and another solute goes to the other side. This is for solutes that your cell needs or doesn't need, depending if it's going in or out, that won't go through the protein channel. I can say it like that. Now, whoa, protein channels and carrier proteins are made of proteins. Never would have known. Oops. There we go. Next, active transport. It goes against the concentration gradient. So the stuff that your body needs, the stuff that's being transported, there we go, goes from low to high concentration, which means they act against the concentration gradient. To do so, they need ATP or energy. ATP is energy, but it's like energy for the cell, a type of energy that cells use. Do I have an example of active transport? Let's see if I can find an example of active transport. Here you go. So we see stuff going into, let's say this is the channel protein for active transport. We see stuff going into the protein, and then here's ATP. ATP goes boom, and it forces the solutes to go out on the other side to where they want it to go. So it forcibly sends a solute somewhere. That's active transport. Next, bulk transport. Bulk transport is the movement of large molecules in a membrane-bound sac, and all membrane-bound sacs in a cell are called vesicles. They move these large molecules the large molecules are moved across the cell membrane. So there are 4 types of bulk transport: phagocytosis, pinocytosis, exocytosis, and endocytosis. First off, phagocytosis. It is the transport of food or just large solids through your cell membrane. So if we look, for example, here, this is your cell. There's a large particle. It wants to enter the cell. Large particle goes to your cell membrane. Your cell membrane folds itself, kind of folds around the large particle, and then it pinches off, and now it becomes a vacuole, a vesicle. It should be a vesicle. I guess you could call it a vacuole. And now we have that inside your cell. That is phagocytosis, transport of large solids. Next, we have pinocytosis. It's the same as phagocytosis, except liquid version. So if you look down here in the right corner, it's a photo of pinocytosis. We have your cell membrane. We have liquid. It goes into your cell membrane. The cell membrane pinches off, and now it becomes a circle floating, a vesicle. Now, to remember the difference between phagocytosis and pinocytosis, think how phagio like starts with a PH, which is pronounced like food. So phagocytosis, food, intake of food. Phagio. My teacher likes to remember pinocytosis by the words pina colada, which is a drink, if you've ever had it. I think it contains alcohol, actually. You might not have had it. You probably haven't. But pinocytosis, pina colada, think liquid. Phagocytosis, food. Okay, other two types, exocytosis and endocytosis. Exocytosis is the process of the large particles, so it can be either the food or liquids. This is the process of them when they're leaving the cell. And then endocytosis is the process of taking material into the cell. So to tell the difference between exo and endocytosis, or to remember them, exocytosis, think exit, so it's leaving, and then endocytosis is just the opposite. So for example, we can have phagocytosis and exocytosis, which is when large solids leave your body or leave the cell. We can also have phagocytosis occur with endocytosis, which is when food or large solids are taken into the cell. Moving on. Quizum. Okay, quiz time. Let's take a look on what I should quiz you on. What did we learn? Okay. What is another name for the cell membrane? Someone's joining. I will pause my recording temporarily. I have resumed the recording because Tony joined for like 2 seconds and then something happened to him. So, Quizum. What did I talk about? Yes, that. The cell membrane, other name for cell membrane. Yeah, I'm gonna annotate because I recently learned that I can do this. And this is really fun. So if you remember, the cell membrane looks like this. Here's some clues. There's a phosphate head. There's some lipid tails. So based on this information, what is the other name for the cell membrane? I'll give you 15 seconds while I draw some more stuff. Okay, the other name for the cell membrane is phospholipid bilayer, which I'm not gonna write out because that's too much work actually. Phospholipid bilayer. Now, this here is a protein channel. What kind of transport does a protein channel do? Does it do facilitated diffusion, regular diffusion, osmosis, or active transport? 15 seconds. Okay, the protein channel, which is, let me switch to a mouse. Protein channel, which is, yeah, this thing. Oh no, my drawing. Anyway, this thing, if you can see my mouse kind of circling it, it does passive transport, where no energy or no aid is needed in having the solutes move through it. Let's go back to Quizum. This here is a carrier protein. What kind of transport does it do? Also passive transport, also diffusion, or osmosis, active transport? Facilitate diffusion, there we go. Which one does it do? It does I think. Wait, facilitated diffusion. F. F for facilitated diffusion. What am I even drawing? Facilitated diffusion. Let's see. Moving on. I'm pretty sure the carrier protein can also do active transport, where energy come. Oh no, I just gave the answer to the question I was going to ask. What is the difference between normal diffusion and facilitated diffusion versus active transport? Tony, what is the difference between active transport and like normal diffusion? Oh, jokes, Tony's busy. Oops. Oh, I drew all over the screen. Give me a second as I erase. Yay. Oh, wait, Tony answered in chat, guys. He says, the difference between active and passive transport is that active transport requires energy, whereas passive transport doesn't, which is correct. Wow, your teacher, one of your teachers knows their stuff. Tony is actually helpful. I was not very helpful for him during the physics unit. Bulk transport. All right. Let's talk. If a cell is taking in, oh no, I just gave the answer in the question. Is exocytosis the exiting of a cell solute? Wait, let me word this better. Exocytosis. What is exocytosis? There we go. 15 seconds before I give the answer. I don't know if that was 15 seconds, but exocytosis is the exiting of stuff from the cell through the cell membrane. Usually exocytosis is performed for your cell to get rid of waste. And then the opposite of that is endocytosis. Other ways of bulk transport we have, if you recall, are phagio and pinocytosis. Now, out of phagio and pinocytosis, which one's the intake of liquids? I'll give 15 seconds before having Tony answer the question in chat. He answered endocytosis. Or your options were phagio and pinocytosis. He answered the previous question, huh? Okay, so I'm gonna reword the question for Tony. Out of phagocytosis and jokes, he answered it, guys. Phagocytosis is the intake or outtake of solids, whereas pinocytosis is the transport of liquids. You have passed the first checkpoint. Let us move on to the next topic. Oh, I gotta erase. Next up, solution concentrations. So there are 3 types of solutions you will hear about: a hypertonic solution, an isotonic solution, and a hypotonic solution. In a hypertonic solution, there's a higher concentration of solutes or solids outside of the cell. Therefore, there's less water. Since there's more solutes outside of the cell, then the solution outside the cell has less water. Because of less water from within the cells, so in your cell there's more water, and the water from within the cells leaves through osmosis to go into the solution, causing your cell to become shriveled. So if we look in the bottom right corner here, we have an image of, for the animal cells, we have red blood cells, and for plant cells, we just have plant cells. Now, the red blood cell, when placed in a hypertonic solution, you can see that it becomes shriveled, wrinkled, smaller. Next up, okay, Tony's here to object to me in chat. Give me a second as I pause the recording. Hi, I am back. Isotonic solution. An isotonic solution is when there's an equal concentration of solutes both inside and outside of cell. Or as I wrote here in the definition, there's a dynamic equilibrium between the inside and outside of a cell. So again, back to the bottom right corner image. Here is a red blood cell in an isotonic solution. Nothing happens to it. It is okay. Now, hypotonic solution. This is the opposite of a hypertonic solution, where in a hypotonic solution, the concentration of solutes is lower outside the cell membrane. Yeah, it's lower outside cell membrane than the concentration of solutes inside the cell. So because there's more solutes, inside the cell, water goes into the cell. So if we come back to the bottom right image, we can see images of a cell in a hypotonic solution, and the cells are swollen. They're kind of fat. Yeah. I should find a better image, a better visual representation of the solutes entering in and out of a cell in different solution concentrations. So, solution concentrations and plant cells. When water leaves the plant cell, because it was placed in a hypertonic solution, causing the cytoplasm of the plant cell to shrink, this is referred to as plasmolysis. So again, if you look in the bottom right corner, in an animal cell, we refer to it, you can honestly just call it whatever you want. You can say the animal cell has shrunk. But in plant cell, the term for it is plasmolyzed. Next, we have turgid, which is when the plant cell is in a hypotonic solution, causing it to be full of water, making it fat. Fat plant cell is called turgid. Next, we have flaccid, which is when the cell is normal, the plant cell, and it is in an isotonic solution. Cool. Let's do a checkpoint. All right. What do you call a plant cell that's fat? You know, when it's swollen from being placed in a hypotonic solution. 15 seconds. A fat plant cell is a turgid plant cell. Now, the opposite of that, what is a shriveled plant cell? What is the term for it? 10 seconds, why not? You guys don't need that long. All right, plant cells are shriveled plant cells are plasmolyzed. Yeah, that's pretty much all we learned in this topic. Tell me what an isotonic solution is. Talk to your screen or mentally say it. You have 10 seconds to figure out the definition. See, I don't know if you guys are actually answering this behind the screen, watching your recording comfortably in your own time. Not gonna judge, at least you're watching the recording. But these exercises, participate. This is are part of a very effective studying method I have once mentioned in the previous lesson known as active recall, which is just quizzing you. It is a good way for you to remember things and recall things for future use. Okay, what was my question again? What an isotonic solution is. An isotonic solution, when an object is in an isotonic solution, it means the concentration of liquid inside the cell and the concentration of the solution, which is outside the cell, is the same, where there's dynamic equilibrium. You guys can remember that word and you can write it down in your science classes to be all fancy and impress your teachers. Next topic, cell membrane. All right, we already know the phospholipid stuff. So if you remember when I talked about cell membrane earlier, I talked about how there's a lot of proteins on the cell membrane, where there's protein channels and there's stuff on the cell membrane and proteins embedded into the cell membrane. Now we're going to talk about them. First up, we have recognition proteins, also known as peripheral proteins. These proteins are on top, usually on the outside of the cell membrane. They act as markers on the cell, which identify what kind of cell they are. They're like passports. So when things pass by, so let's say white blood cells are passing by, and then they're looking for viruses or diseases that don't belong. And then they see this cell of yours, and then they check it with its marker. They look at it. Obviously, they don't look at it, but this is metaphoric. They look at your cell, and they're like, oh, this is a marker. You are part of us. You're one of us. You're good to go. Now, if they look at a cell, and this marker, there's something wrong with the cell because there's no marker on it, or its markers are weird, then the white blood cells will attack it because it is a foreign thing that is not meant to be in your body. Now, the thing about recognition proteins, sometimes the white blood cells actually mess up, and they'll look at stuff that is okay in your body but mistake it as foreigners. So if you're, for example, let's say you have allergies, pollen. No, let's say you're allergic to fish, and then you eat fish, and your body sees these fish cells or something, all right? Normally, your body is like, oh, this stuff is safe. It won't kill me. But if you're allergic, your white blood cells are like, they see the markers and they're like, this is wrong, this is bad, and then they start attacking it. That's when they mistake your recognition proteins incorrectly. Now, one example of recognition proteins we know often about is blood types. You have A-type blood, B-type blood, AB or O-type. The different blood types are identified by the different recognition proteins on them. Moving on, receptor proteins. Receptor proteins bind to certain molecules to bring them into the cell by endocytosis. What? Viruses are, okay, there we go. Receptor proteins. So let's draw this stuff out. You have a cell. You have a receptor protein. Let's say there's like, let me think. There's something that wants to go into your cell. Let's say a large solid. You know phagocytosis? Yes, phagocytosis. Let's just say there's a piece of food. All right. This piece of food. Oh, I know what's a good example. A hormone. So hormones, they do a lot of stuff to your cell. They impact your nucleus and they tell your nucleus to do stuff to the cell. Anyway, hormones will bind to the receptor proteins, and receptor proteins like, okay, this is something that's safe to intake. And your receptor protein basically lets this hormone in, lets this object into your cell. And you're good to go. Now, sometimes these receptor proteins get tricked by viruses. So let's give a nice example that we have all heard of, the coronavirus. I actually got tested for it today. I'll tell you guys if I test positive. Anyway, the coronavirus binds to your receptor protein. And normally for foreign objects, your receptor protein will know and it won't let the stuff in. Oh, Tony asks if I'm quarantined. We can talk about that later. Okay. The virus tricks your receptor protein into letting it in, even though it's harmful to you. Your receptor protein like kind of gets confused, and then your coronavirus, yes, it goes into your cell, and then it's bad for you. You get sick, you cough, you have a fever, and then you might die. Stay safe. Wash your hands. Tony, I will answer your questions about why I took a COVID test later. He is talking in chat. Next up, membrane proteins and treatment. So a lot of you guys might have already heard of HIV, which is human immunodeficiency virus. Again, if you're, as I said in the previous slide, viruses bind onto receptor proteins. Receptor proteins? Yes, receptor proteins, and then they trick them into letting them into the cell. So one proposed way of using what we know about cell membranes to prevent HIV, human immunodeficiency virus, is what's known as a lock and key scenario, where they kind of just block the attachment site or the receptor proteins, so that HIV can't attach to that receptor protein, which is kind of weird, not gonna lie. Like, so much work blocking the receptor proteins. Anyway, not important. Next up, cancer. We need more. So there has been proposed ideas on using kind of a way to using receptor proteins and maybe manipulating these receptor proteins as a treatment for cancerous cells to kind of treat cancer. But at the moment, what our treatment for cancer damages both healthy and cancerous cells. Let's go to the next slide. Tony, stop asking me why I wasn't at school today. COVID testing. It's because of COVID testing, because I was doing COVID tests instead of going to school. Membrane technology. So this is expanding more on the previous slide where we talked about how we can use what we know about cell membranes to use as in medicine, for medicinal uses. Now, one treatment we've made from our knowledge of cell membranes is liposomes, which are synthetic membranes that mimic natural cell interactions. Essentially, scientists have been able to make fake mini cells, fake vesicles, man-made vesicles that can go into your cell, that bonds with their receptor protein, and receptor protein's like, okay, I will let this thing into my cell, into me, the cell. So liposomes are a fluid-filled sac surrounded by a man-made phospholipid bilayer. They are sometimes used in drug delivery medications, and sometimes they have DNA inside them. The liposomes attach to infected cells and deliver the cell, like the medication that is inside of it, via endocytosis. Sometimes, as medicine, they put DNA inside the liposomes, and the DNA, I think, tells the nucleus how to destroy what it's trying to treat. Wait. Okay, there's something wrong with your cell, right? That's why we're delivering medication. And sometimes they put DNA in it. DNA goes into your nucleus, and your nucleus, this DNA tells your nucleus how to fix the problem with your cell, essentially. So fixing it from inside out. Checkpoint. What should I ask about? Let's take a look. Give me an example of when recognition proteins are used. Wait, no, that's a weird question. That's kind of a badly worded question. Here, what is the role of recognition proteins? It's kind of in the name. 10 seconds. All right, recognition proteins. Same with the deal. They are proteins that are on the cell membrane, and their role is, well, you can define them in any way, as long as it's similar to what I'm about to say, which is they're just identity markers of the cell. All right, what is one way, so I've given, I think, 3 reasons or examples in the previous slides. Just list one of them. What is one of the ways that we've utilized what we know about cell membranes in medicine? 10 seconds. Let's go back to the 3 examples. 1, HIV, lock and key scenario, they block the attachment site or the receptor protein that's associated with HIV. 2, cancer. This is honestly awkwardly worded. I have no idea what I'm trying to say here. And then liposomes, which is man-made vesicles that go directly to your cell. Man-made vesicles that contain medicine. Next topic. Ooh. We kind of already did a break. Whatever. Next topic, applications. Hormones. So this is like how in the earlier slide I gave applications of what we know about cell membrane. This is more applications, essentially. First off, hormones. Hormones, here, let's do some drawing. So, if you remember, when I talked about receptor proteins, I mentioned hormones can bind to receptor proteins, and then they make your cell do stuff after going into your cell. Now, one hormone, insulin, which is a protein hormone that's produced in the pancreas, stimulates your cell to take in glucose. In someone with diabetes, or more specifically, diabetes mellitus, type 1 diabetes mellitus, they are unable, the ability of their cells to take in sugar is kind of bad because your pancreas isn't secreting insulin, the hormone that helps your cell take in glucose. Your pancreas isn't doing its job, one of its jobs. Your pancreas has many jobs. Anyway, insulin, sometimes because of this, we can inject insulin. One type of way to treat type 1 diabetes mellitus is by injecting insulin into your cell. We can now make man-made insulin. And then the insulin binds with your membrane receptor proteins, it goes into your cell, and then your cell can take in glucose again. In the past, when man-made insulin wasn't available, I don't remember exactly, but I think my teacher said that they kind of took insulin from like cats or like lamb or sheep or something. Anyway, at some point in time, they couldn't make man-made insulin, so they just took insulin from something else, another living thing. Next, dialysis. Dialysis is a treatment of kidney malfunction. If you guys should know, maybe not, your kidneys, they look like beans, look like this. They are about the size of your fist. And they filter your blood to create urine, pee. So again, the kidney's job is to remove waste products and to remove excess water. It also maintains your body's chemical balance, so by removing stuff. Dialysis, when your kidneys malfunction, your body is unable to properly remove these waste products and remove any excess water. So, the treatment. There are two types of dialysis for treating kidney malfunction. First up, peritoneal dialysis. This is like, peritoneal dialysis. So the two types of dialysis, one must be done in the hospital and one can be done at home, kind of. So peritoneal dialysis, you go to the hospital, I think you have surgery, they do some stuff with you, whatever, you go home, and then you can do the rest of the procedure of peritoneal dialysis at home. Now, the peritoneal dialysis uses your peritoneum, which aligns in your abdominal cavity. Your wastes, which is in your blood, they pass through your peritoneal cells, or also known as they pass through peritoneum, okay? They pass through peritoneum. And then the waste or toxins from inside your blood kind of diffuses or filters through your peritoneum and then into an external solution we call it dialysis solution. So here on the top right image, you can see we have your peritoneal cavity, and then we have your dialysis machine. So if you have peritoneal dialysis, at the end of each day, you kind of attach the dialysis machine to yourself, and then your blood just filters through it, cleans itself, and then you detach it, and then you're good. You're good for the rest of the day. Yeah. In summary, it removes waste from blood through a layer of tissue in the abdominal cavity, the peritoneum, by doing a solution exchange. You could just say through diffusion, the end. That's all you need to know. Next, hemodialysis, second type of dialysis. This has to be done in a healthcare facility. First, let's erase my drawings of a bean kidney. Okay. Hemodialysis requires constant monitoring. In hemodialysis, your blood is filtered through a machine that uses diffusion. So in peritoneal dialysis, we use your peritoneum as a sort of filter. In hemodialysis, we have a machine to filter you. So you have to go to a hospital, and then they shove this needle up your arm, and your blood goes through that needle, and then you have to sit around for a while, waiting for all your blood to circulate through your body into that machine, be filtered, and come back into you. Okay, now we talk about reverse osmosis. If we remember, osmosis is the diffusion of water. And water, since water is special, it has its, instead of saying diffusion of water, we just say osmosis. Water is very special in science. Anyway, desalination is a process of removing salt from a solution via reverse osmosis. So to do so, reverse osmosis is performed by having a solution be pumped through progressively smaller semi-permeable filters. This process requires a pump/energy because it's going against the concentration gradient. So if you look here, down here, we have the reverse osmosis process in this image. On the left, we have unfiltered water, and on the right, we have distilled water, which is filtered water, pure water. Did you know, guys, pure water is actually bad for you? Do not drink pure water because it's so pure, your body's like kind of not okay with it. It's too pure for your body. It's bad for you, pure water. So don't drink pure water. Drink non-pure water, impure. Okay. Back to my diagram. Unfiltered water, we have applied pressure, pushing on it, and it pushes the solution through our layers of semi-permeable membrane where the holes inside it gets relatively smaller and smaller, where all the contaminants and stuff we don't want get stuck, and only water molecules are able to pass through the semi-permeable membrane. So we have purified water. Quiz time. Checkpoint. What are the two types of dialysis? No, let's talk about what dialysis is. What is dialysis? Dialysis is a kind of a treatment. Okay, as long as your answer is, again, something along these lines, you're fine. So a treatment for when your kidneys are malfunctioning. So, multiple choice question. Which type of dialysis requires constant monitoring and must be done in a healthcare facility? Peritoneal dialysis or hemodialysis? Hemodialysis requires you to be in a healthcare facility where you have a machine that does your monitoring, does your filtering externally. Our next topic, cell structure. First up, why are cells tiny? If you look here in the bottom of this image, oh, all right. Do you guys remember in maybe a lesson some time ago, which is 1 week ago, the lesson from 1 week ago, I talked about how, I think it's 1 week ago, like in bio, things like to have a really high surface area, where a lot of things go out of their way to maximize surface area. Now, looking at this diagram, we can see that when objects are smaller, their surface area to volume ratio is larger. So here, the surface area to volume ratio is 6:1 in a 1 centimeter cube. But in a 3 centimeter cube, the surface area to volume ratio is only 2:1, where the surface area is only 2 times that of the volume. But in a 1 centimeter cube, the surface area is 6 times the volume. All right, coming back here. So the reason cells are tiny is because when they're smaller, they have a larger ratio of surface area to volume, so they can maximize their surface area. Which is something everything in bio wants. Every other thing in bio wants to do, maximize their surface area. By maximizing their surface area, there is a greater efficiency for materials to enter and exit the cell. So there's a larger surface area, more things can come in at once and leave at once. And because there's a smaller volume, when materials enter the cell, they can get to where they need inside the cell pretty fast. They don't need to travel very far since it's pretty small inside the cell. And objects and waste leaving the cell leaves pretty fast since there's a very small area to travel. All right. So we humans are multicellular organisms. Any living organism we see is multicellular because unicellular organisms cannot be seen by the naked eye. Now, multicellular organisms, the advantage of having many cells, why we evolved to have many cells, is because in multicellular organisms, there is a division of labor. Each cell has its own function. For example, you have liver cells and say stomach cells. Your liver cell does liver stuff. Your stomach cell does stomach stuff. Each cell has its own function. They do their own thing. And these specialized cells for each of their own functions are more effective and efficient in their role. So a liver cell does liver stuff very effectively. But if you put a stomach cell and said, do some liver stuff, stomach cell's like, no, I can't, bro. So by having our specialized cells, we have very effective and efficient cells for their function. There we go. We also have cell interdependence. So you have many cells in your body. if one of your cells, let's just say you get a cut, and some of your cells kind of get hurt. Some of your cells die because you got a cut. You're not going to die from your cut. It is okay when a few cells die. And your individual cells don't matter. Individuals don't matter. It's the group that counts. Communism. Anyway, despite specialized cells, they share DNA. So even though we have different kinds of cells, so let's go back to our liver and stomach cells. Your stomach cell is actually able to perform liver cell functions because in the end, the composition of cells are pretty similar. They have a nucleus, they have centrioles, blah, blah. So they share DNA, but it's just not as efficient. However, the thing about our cells is that when we are born, we have lots of stem cells, which is like baby cells that can grow. There are neutral cells that as you grow older, they become cells. They become specialized cells. Stem cells can develop into liver cells, they can grow into stomach cells, they can grow into any cell. They develop. I think they're usually found in babies as your baby is developing. Yeah. So basically your cells share DNA. Next, multicellular organisms allow for efficient exchange of materials with their internal transport system. So here, your blood, what's it called? Your circulatory system, which is your blood vessels and all that. We have internal transport system, which is our blood vessels, and we have an efficient exchange of materials as our blood vessels carry oxygen to all the cells in our body, and then it brings CO2 to our lungs, and we breathe in and out. We have this efficient intake of oxygen and outtake of CO2, which is the exchange of materials, with our internal transport system, which is the circulatory system. Unicellular organisms. In unicellular organisms, such as bacteria, one cell performs all the functions, since it's only one cell. We are multicellular, we have multiple cells doing different functions. In a cellular organism, that one cell does everything for itself. It eats, it digests, it breathes, it does everything. It is restricted by the rate of diffusion when it wants to exchange materials. So, for example, let's see. I don't know how to give an example, but compared to multicellular organisms, where you have specialized systems, transport oxygen, like blood, unicellular organisms kind of just have to wait to get stuff. I don't know how to explain it. I hope you kind of have an idea of what this is supposed to mean. Now, in unicellular organisms, when one cell dies, because there's only one cell, it's gone. It's dead. The organism is dead. Bye-bye. It is, yeah. It is erased from existence. Now, classifications in multicellular organisms. First up, as I said before in cell theory, cells are the smallest units of life. And then when cells group together, a group of similar cells, they come to band together, they are referred to as tissues. So you might have heard of like tissues in your body. And now a group of tissues, so let's say a bunch of liver tissue, they combine. I want a fancy word for combine. What's a fancy word? I can't remember it. It's on the tip of my tongue. A group of tissues combine to form an organ. Now, a group of organs working together is referred to as an organ system. And then different organ systems working together creates an organism. Let's see if I have an image. Here we go. This is a nice image. Is it? Yes, it is. Here we have a muscle cell. And a group of muscle cells creates muscle tissue. Muscle tissues combine together, creates an organ. In this case, it is your bladder. And yes, your bladder is made of muscle tissue. And then your bladder, combining with other organs such as kidneys, working with other organs, creates an organ system, in this case, your urinary system. And then if the urinary system works with other systems, your circulatory system, your respiratory system, it makes you a human being. How unfortunate. All right, here's a quiz. Why are cells tiny? Okay. Cells are tiny because that way they have a higher surface area to volume ratio. Through this, they are more efficient in their exchange of materials. I'm going through this stuff way too fast again. Like all the information in this lesson. Anyway. Why are multicellular organisms better than unicellular organisms? In truth, they're not, but for my sake, and because I am biased, why are multicellular organisms better? Give one reason. Actually, you know what? Give two. Okay. Multicellular organisms, when one cell dies in your body, you're totally fine. You're not gonna die. You're all good. Another reason, you have efficient functions. So you have specialized cells for functions in your body. And therefore it makes it more, your specified functions are more efficient in that way. Whereas in a unicellular organism, it kind of tries to do everything. Think about it this way. It's like for our specialized cells, we have majors. Okay, your liver cell majors in liver stuff. Your stomach cell majors in stomach stuff. They went to school and they learned stuff. They went to school, they studied it, and they became very smart in their area. And a unicellular organism, it kind of learned a bit of everything and was like, I think that's good. Now I know everything, and knows kind of like surface area information. So it kind of does everything, but not very well, since it doesn't know that much. Whereas your specialized cells in your body, they don't have as many functions, but the function that they do perform in a way is more efficient and much more effective. Plant tissues. Now we're talking about plants. We're done talking about your body. Oh. All right. 3 types of tissue in plants. Dermal tissue, which is like the skin of the plant, also known as the epidermis. And then we have the ground tissue, which is the layer beneath the epidermis. And then we have vascular tissue, which is transport tissue. Now look here on this image. This is a cross section. This is like, if you cut this plant right here, across the line in half, and then you looked up its stalk through a microscope, this is the outside of the cell. The outside of the plant. This is the dermal tissue, the skin of the plant, the epidermis. Inside of that, we have the ground tissue, which is a layer just right beneath the epidermis. This is the layer that performs photosynthesis. Cells in the ground tissue, you'll learn this again later, have a lot of chloroplasts. This is one of their specialized functions. So they are more efficient. Again, they are a specialized cell for photosynthesis. So they have a lot more chloroplasts than any other cell in the plant. And then inside of the ground tissue, we have the vascular tissue, also known as the transport tissue, which includes the xylem and the phloem. Phloem. You guys probably learned this since grade 6. I swear I remember learning enzyme flow in like grade 5 or 6. I think it's grade 6. One of them transports water, one of them transports solids. I don't remember which does which, but we'll learn that again later on. Now, the dermal tissue. Now coming back here, it is 1 cell layer thick. There's only 1 cell thick, 1 cell around the dermal tissue. Kind of. No, there's more than 1 cell, but as you can see, there's like 1 cell in terms of its width. It does the exchange of matter and gases in and out of the plant, so it acts like the cell membrane of the plant. If you remember, it's a skin / epidermis. So, and it's semi-permeable. So something that these cells let in is sunlight. By letting sunlight into the cell, they can perform the ground tissue can perform photosynthesis. It is the part of the shoot and root system in the cell. And you will learn about that, the shoot and root system, in the next lesson. Goodbye. Goodbye. Have a good weekend. And show up to my next lesson. Thank you. Would really appreciate you all showing up. Goodbye. Bye-bye.